Welcome back. We have in the last session we ended up by saying that the angiotensin converting enzyme is a very good target for inhibitor design, and if you can design an inhibitor and find out a very good inhibitor with the appropriate properties which include ADME properties and PD properties, toxicological properties, then you can have a molecule which can be sold as a drug, but between that there is this clinical trials. Okay. Now, ACE, I told you that based on the carboxypeptidase A structure, that it was assumed that ACE will have a very similar kind of active site geometry, except that it is a dicarboxypeptidase. So, you have to shift the zinc which is binding to the carbonyl and assisting the hydrolysis. You have to shift the position of the zinc little bit away from the carboxy end. Okay. I have shown that this is the rough sketch of the active site geometry. Now, I ended that time by saying that you need a clue from somewhere which is your starting point. In this case also it goes like that in 1965 one person Ferreira, he reported that mixture of peptides from venom of a South American pit viper, this Bothrops zararaca. So, that is it's a name, biological name for a particular type of snake, and the picture of the snake is shown here. Okay. It is a deadly poisonous snake. So, from the snake venom, this person isolated a mixture of peptides, and they found actually how many peptides? 9 active peptides were isolated from this venom, and the structure of the out of these, the structure of a pentapeptide PYR, lysine, tryptophan, alanine and proline. PYR is what is called pyroglutamic acid. So, glutamic acid which is cyclic, this is what is pyroglutamic acid. So, it has got a strangely it has got a pyroglutamic acid. So, pyroglutamic acid, lysine, tryptophan, alanine, proline, this pentapeptide was identified. Okay. The pentapeptide was shown to inhibit the conversion of angiotensin 1 to angiotensin 2 and bradykinin degradation in vitro and also in vivo. Okay. So, this was your starting point. The clue was obtained from the venom of this snake, and they isolated the peptide. 9 peptides were isolated, and out of these a pentapeptide whose sequence is shown here was found to be very good in inhibiting the conversion of angiotensin 1 to 2. That means vasoconstriction will go away, and it also does not destroy the bradykinin, causes less degradation of bradykinin, which is a vasodilator. Apart from these 5, the structures of 6 more peptides were determined by other persons, Ondetti and co-workers. Now, the peptide with the greatest in vitro activity was the pentapeptide, as I already mentioned, but a nonapeptide, they have also isolated a nonapeptide which is having this type of sequence pyro-TRPA, proline, arginine, proline. That means, up to this point is that pentapeptide, and then there are 4 more amino acids called a teprotide, had the greatest in vivo potency and was effective in lowering of blood pressure. So, lot of peptides were isolated, and they found that some of these peptides have very good potency in inhibiting the angiotensin converting enzyme and thereby reducing the problem of hypertension, lowering the blood pressure. Okay. However, these peptides, the problem is peptides cannot be drugs because of the problem. The problem is that peptides, before they reach the target, it will be hydrolyzed by many peptidases that we have, which are unspecific, non-specific. So, they hydrolyze the peptide before it reaches the target. So, peptides are very difficult to be utilized as a drug. So, although you know that these peptides have very good activity, but you cannot use them in practice. Okay, now, let us again come back to that pentapeptide, the pentapeptide that was that is another, this is glutamic acid, lysine, tryptophan, alanine, proline. So, what it says IC50 values, IC50 is that the 50% that to lower the enzyme activity by 50%, the amount that is needed to do that is called IC50. Okay. So, they are making these type of peptides which are very similar, because if I go back to the earlier one, you see they all ended, this was a proline at the terminal end. So, they do not disturb that much. What they have done is only pyroglutamate, they are putting a glutamic acid. Okay. So, this pentapeptide has a IC50 value very good, 0.05 microgram per cc, and then if you take, instead of tryptophan, if you take phenylalanine here, then also the value remain the same. That means, at this site, if there is a binding pocket, that recognizes only this aromatic amino acids like tryptophan and phenylalanine. Okay. So, then they started, this is what is called structure activity relationship. They are slowly taking one amino acid off, like the glutamic acid if you remove. So, you get lysine, tryptophan, alanine, proline, that means the first, based on the first peptide, and that has got, now the IC50 value got increased a little, 1.2. That means, glutamic acid gives a better one, but if you take IC50 value is increased means actually it is becoming less potent, the value is 1.2, that is still not too bad. And then if you remove the lysine, then you have phenylalanine, alanine, proline, that has got 1.4. That means, there are some minor roles of this glutamic acid, of this lysine, and these two. So, it becomes 1.4, but when you change the last one, this one, the third one, phenylalanine, you get alanine and proline, but now you see the IC50 value 50. So, IC50 value has increased to a large extent. So, this is a very bad inhibitor. Again remind, IC50 higher means less potent inhibition. Okay. Now, what happens, at that time it was known that a compound which is called benzyl succi, it's called a substrate which is a phenylalanine based peptide. Okay. This is what the carboxypeptidase, because carboxypeptidase I told you that it recognizes, it likes the aromatic amino acids at the end, although it hydrolyzes aliphatic amino acids at the end. So, if I take the aromatic amino acid, say phenylalanine, carboxy, then an NH and then CO2. I think I gave you this active site geometry earlier. So, I start from this carboxypeptidase. So, this is a substrate. Now, this is a substrate and this is hydrolyzed. That means, the carbonyl is hooked to the zinc to activate this carbon. The phenylalanine has a hydrophobic pocket here. So, that goes and binds, and the carboxy must be binding to a plus charged basic amino acid. In this case, it is arginine. Okay. That is for carboxypeptidase. Now, someone found that if you remove this NH and forms a CH2 here, okay, and also remove the carbonyl, and instead you put the carboxylic acid here. That means, there is no peptide bond after this, and this peptide bond is replaced by a CH2, COH and then CH2. So, what will be the effect of this? First of all, this is a sessile bond, means which can be broken, it is a peptide bond. But now what you have done, instead of the peptide, you have made a carbon-carbon bond which is not sessile. Sessile means you cannot break this. Now, this is what is called a peptidomimetic, means you are not having a peptide, but peptidomimetic group. Peptidomimetic group is used just to stop the hydrolysis. Okay. Now, what will be the, this was found to be a very good inhibitor of carboxypeptidase. So, that means why it is very good inhibitor? It has got the CO2 minus, so which is bound to this positive charge by electrostatic interaction or a salt bridge, and then you have this phenyl group. So, the phenyl is having stabilizing interactions here, and then what you have? You have a CH2, so no NH, and then you have a CO2 minus. Basically, in the biological system it will be present as this CH2, and then you have O minus. So, basically this is a delocalized CO2 minus, C double bond O minus. So, I can say a delocalized negative charge. Okay. So, that will be what will happen. Actually, in this position there is this zinc plus. So, now that will form a chelate to the zinc ion. Okay. So, basically what you have done, you have replaced the carbonyl with a CO2H. The CO2H is now the ligating to the zinc, because that is well known, like say EDTA, you have this CO2H. So, that chelates to the metal ions. So, you can have chelation to the zinc, instead of NH you are putting CH2. So, no hydrolysis will take place. This phenyl is still there, that binds to the pocket, and the CO2 minus is also here, that binds to the pocket. Okay. So, this is how, what is the mechanism of this benzyl, this is called, now this is called this is phenyl, this is not actually phenyl, oh sorry, this is benzyl succinic acid, very simple compound, benzyl succinic acid, because this is not phenylalanine, because you do not have NH here. So, succinic acid is CO2H, CH2, CH2, CO2H. So, you have benzyl succinic acid. So, it was found that benzyl succinic acid is a very good inhibitor of carboxypeptidase. Okay. So, now from these observations that these peptides from the snake venom are very good inhibitors, and this benzyl succinic acid is a very good inhibitor of carboxypeptidase. So, that gave the scientist a clue how to design an inhibitor of this ACE enzyme. Okay. So, what they do or what they did, let me again show you. Interesting point is all these peptides, the terminal, the C terminus is a proline. Okay. C terminus is a proline, that and then it is alanine. Okay. Then it is alanine, that means these are obligatory amino acids if you want to design an inhibitor of ACE. So, what people have done, they made lot of derivatives of proline, like this is a proline. See, that peptide was what? The starting, the snake venom peptides were proline, and then you have an alanine, that means N, CO, CH, CH3, this is alanine, and then remember, if you remember, there was an aromatic amino acid here, CH, and then NH, then CO, then there is an aromatic amino acid like tyrosine or phenylalanine, that is in the peptide that were isolated from the snake venom. Okay. So, if you think that, if you based on this, that means if you want to make an inhibitor, you should not change the proline, because proline, all the peptides ending up with proline as the C terminus. You should not change the alanine also. What you should do, this peptide should be, now that benzyl penicillin has given you the idea that that should be CH2, so that this cannot be broken now, it is a carbon carbon bond. Okay. Now, based on this, people have started working on that. What you have to do? You have to make a proline, then you have to take an alanine and then take a CH2 alanine, it is not alanine, means CO, CH2, then a CH2, that means a succinic acid derivative here, and then you have to that phenyl group should be the ideal. So, people started working on this. First they had this proline, then CO, as I said, then they did not take the alanine. They first thought that methyl may not have much effect. So, remove the methyl, okay, and then a CH2 and CO2H. This was the first inhibitor that started. This was your starting point. They saw that IC50 value is very bad, 330. Then they started putting the methyl. Okay, they put the methyl here. Once they put the methyl, it becomes 22. That means methyl is very important, and the stereochemistry is also very important. If the methyl is alpha, it has to be L-alanine. If it is methyl alpha, means it is not alanine, it is the succinic acid, but I am just comparing with alanine. So, basically when the methyl is beta, you have activity, when the methyl is alpha, you have 1480. So, that means the stereochemistry is so important, which is absolutely required for enzyme, any enzyme inhibition or enzyme catalysis. Okay. So, this is the, so up to this point you cannot change now. Then you have CH2, CO2H. So, you did not have any peptide bond here. This CO2H is the one which chelates with the zinc, that was the idea. So, then they started alpha methyl, then reduce the, then they have CH2, no methyl, again remove the methyl and then had a sulfur here, because we know that sulfur also is a group which can form SCH2, suppose CH, say methyl, and then the proline, because sulfur is also a group which can have a very good chelation to the zinc. Okay. So, they started putting sulfur. So, first 1 carbon sulfur, then 2 carbon sulfur, then but they did not have the methyl yet. So, you see the values slowly going down as they put the sulfur, 0.20, before that 1.1, when 1 carbon spacer is there, then 2 carbon spacer is there, they found 0.20. So, first they optimize the number of carbon atoms attached to the sulfur, 1 is 1.1, 2 is 0.2, 3 is again 9.3. So, that means these 2 carbons are the ones which you require, okay. And then they put the methyl now. So, then they put the methyl and this is the compound, and this has got a very good IC50 value, 0.02 to 0.08. This is what is the drug, and if you make alpha methyl again as expected, it will go up. So, this is the one compound which was first, which was approved by the FDA, and this compound make a lot of interest, creates a lot of interest in this type of research. And you see, this is the first example of what is called the so called rational drug design, because everything started from one clue and then comparing with the existing information, and slowly by structure activity relationship they ultimately came out with a compound which is called captopril. Okay. So, captopril is the first ACE inhibitor which was introduced into the market in the 80s. Okay. However, that is not the end of the story. Every drug may have some side effect. Okay. Like captopril, the problem is the sulfur. Because of the sulfur, it creates a lot of problems. What are the problems? The problem is basically, where is the problem of captopril? I think something is written here. Use of captopril, I am not going into these details. The captopril's effect was rashes, that allergic reactions, because sulfur allergy is there, like penicillin I told you, and then loss of taste. So, people lost their appetite, loss of taste, so they are not eating much. So, this is probably due to SH groups, and however, if you withdraw the drugs, then these are reversible effects. If you withdraw the drugs, then again you get back your taste and the rashes also go out. So, captopril is not the ideal drug, they realized that because this is one important issue, that antihypertensive drugs is not that like antibiotics, that I take a course of 7 days and then withdraw, but here you have to take almost lifelong. So, if you have a loss of taste lifelong, so what you can think that it is a miserable state of life. So, captopril is not the ideal. So, then started removing the sulfur and coming back to removing the sulfur and then see whether a better design can be done. Okay. And this is, I think, here one slide is, yes. So, remember captopril is, there is a methyl here, beta methyl, then CH2SH. Now, they were again back to the carboxy, because carboxy is the one like benzyl, that is the benzyl succinic acid was the inhibitor of carboxypeptidase A. So, they again went back to the carboxy, because carboxy also creates chelation with the zinc. Okay. So, they started doing the structure activity relationship. What they did, different type number of carbon atoms, then put this NH and CH2, peptidomimetic chemistry, and then, so after all these, doing all these, proline, alanine, CHMeCO2H, everything. So, you see there is an increase, there is a decrease in the IC50 value, and finally, they ended up by making a compound, which is this. Now, I write this compound. See, what is that? You have a proline. If you have a proline, that means this CO2 minus. Of course, proline is obligatory, because you have to have this electrostatic then they didn't change the alanine, methyl NH, alanine, and then methyl NH, up to NH is alanine. Then alanine is not connected by a peptide bond, but a CH, because you cannot have a peptide bond, because that will be then hydrolyzed by the angiotensin converting enzyme. So, CH, and then actually initially they have this compound CH2 CO2H. Okay. This CO2H was basically, you can write in this fashion, not here, you can write the CO2H as a chelating group. So, you write CO2 minus like this, and this is the zinc. So, this is where the zinc is chelated. There is a pocket here methyl, there is a pocket here for the carboxy. Okay. So, that is how it goes. Now, let us see where is this compound. This compound is this is the CO, then CHMe alanine, then CH2. So, this compound is basically this one, whatever I have drawn. This has got an IC50 value of 2.4, but that is not very good. IC50 value is less, should be less than 1, 0.00 something or 0.0 something. So, this was this, because you see IC50 value will ultimately determine how much compound, what is the dose of the compound you have to take. So, higher IC50 means higher dose of compound you have to take. Higher dose means you have to, you ran the risk of crossing the therapeutic index. So, lot of issues are there. So, you have to lower it further. Okay. So, then people realized that in the carboxypeptidase, or if you look at the peptides again, let us go back to the snake venom peptides. I think that will give you a very good idea. Snake venom peptides, where is that? Here. This is the proline. So, you have utilized the proline in your design. You have alanine, you have utilized the alanine also for your design. The third one means on this side is what is called an aromatic amino acid. So, what you have done? You have exploited the binding of the carboxy of the protein the proline, you have exploited the binding of the methyl, and these carbonyls also have some hydrogen bonding interactions. You have exploited the binding of the zinc by utilizing a carboxy here, but what you have not done is exploitation of this binding pocket, that is not yet done. So, in order to get a better inhibitor now, what you have to do? You have to exploit, you have to additionally put this aromatic ring, so that it goes and binds to the pocket. Now, here one nomenclature system I should tell you, that if some protease hydrolyzes this peptide bond, then the amino acids on the N terminus side, that means the first amino acid, suppose this is ALA, that will be called S1 pocket, where alanine, and then if you go on to the N terminus side further away from the actual peptide bond which is hydrolyzed, so then you have an S2 pocket. Then you have on this side it is S1 prime pocket, then S2 prime pocket. So, if I say that in case of say this angiotensin converting enzyme, in the S1 pocket there should be an aromatic amino acid. Is that correct or not? So, we have to see that the hydrolysis usually takes place, it is a dicarboxy peptidase. So, it will be the hydrolysis of the bond between the tryptophan and the alanine. So, S1 pocket is which one? So, S1 pocket will be the S1 pocket is the tryptophan. Let me see that it must be given somewhere, this type of nomenclature. Yes. See here the peptide bond that is hydrolyzed is this one. Okay. This is called the S1 pocket. So, in the S1 pocket what you have is phenylalanine or tyrosine, the aromatic amino acid. So, if there is another one here, that will be S2 pocket. So, the normal numbering goes to the N terminus side, and on the C terminus side you have S1 prime pocket and S1 prime pocket. So, proline is at the S1 prime pocket. This is all in relation to the peptide bond that is being hydrolyzed. So, that is S1 prime pocket, this is S1 pocket, this is S2 pocket. So, what you have not utilized is basically that S1 pocket has not been utilized. You have utilized these pockets, you have utilized, but you have not utilized the other pocket. See, the reference is the peptide. So, on the right side is S1 prime, then S2 prime, second amino acid, third amino acid is S3 prime, and on the left side the first amino acid is S1, the second one is S2. So, in this case what you are hydrolyzing is, okay, here it is also you can say that the hydrolysis of this bond, because it is a dicarboxy peptidase, that means this goes off, a dipeptide. So, your S1 will be which one? This one. S1 prime will be which one? This one. Okay. S2 prime will be which one? Is this proline. See, what you have is a proline, you have alanine, you have an aromatic amino acid. So, the bond that is being hydrolyzed is here. So, this is S1 prime, this is S2 prime, this is S1. I hope this is clear. Now, let us again go back. So, we have not, or these scientists have not utilized the that S1 pocket yet. So, that has to be utilized. So, what they did now, in order to develop this enalapril, I think the chart is somewhere here. In order to develop enalapril or enalaprilat, what they did, they have a proline, they have alanine, they have this CH, and then on the CH they have this CO2 minus, which is a chelation to the zinc, and now you know that here there is a pocket for the aromatic compound. So, now they put a CH, what is that, CH2 pH, because that they had to monitor that how many carbons are required to get into the phenyl, CH2, two CH2s, and then there is a pH. So, I should write this way. So, now there is a pH here. So, they must have actually monitored it. This is a structure activity. First they attach direct pH, then they attach CH2 pH, then they attach CH2 pH, and then found that this is the compound which has got, this is the same compound. This is what is called enalaprilat, which has got an IC50 value is 0.0012. Okay. So, this ultimately became the drug. However, there is still some twist left in the discovery process. What they found, once you have an inhibitor, you have to do the PK/PD studies. PK is basically ADME studies, absorption, distribution, then metabolism and excretion. This drug unfortunately is quite polar and it was not absorbed properly from the gastrointestinal tract. Okay. So, if that be the case, you are not getting the correct concentration of this enalaprilat in the bloodstream. That means, it was not working as it was intended. That means, in vivo activity was much less because it was not absorbed. It has got very poor absorption property because it is quite polar. So, what will be the answer? You make it less polar. So, what they did, this carboxylic acid which was chelating to the zinc, they made it as the ester. If you block the carboxy as the ester, it loses its potency to, it cannot now chelate to the zinc, that is not possible if you make the ester. As long as it becomes carboxylic acid, then it can do that. However, when you put ester, it has become more lipophilic now. I told you about the log p value. So, it satisfies the log p value if you put the ester. Okay. So, now it is absorbed from the gastrointestinal tract. It is a good it has got good absorption property, and the beauty is that as soon as it goes into the bloodstream, there are many non-specific esterases in the body which hydrolyzes it and forms the enalaprilat in the bloodstream. So, basically this is what is called a prodrug. This is not the drug, this is a prodrug. This is has got very good your pharmacokinetic property. So, it goes, distributes, absorbed and distributes, and then the non-specific esterases that are present in the blood. So, they are going to hydrolyze it and converts it into this enalaprilat, and now the enalaprilat acts as a very good antihypertensive agent. Okay. So, now there are new generations of further generations. See what happens, enalaprilat, then you can get even, they tried to make a even better one. There is ramipril is a drug which is used also similar times, I think, but I know enalapril has been prescribed to many patients these days in order to manage the hypertension. Only before I stop, I touched the point a little bit that you can also target the angiotensin 2 receptors, because angiotensin 2 exerts its effect by binding to its receptors. So, if you can use antagonist of these receptors, that also will act as an antihypertensive agent, and in fact, there are drugs which are called angiotensin 2 receptor antagonist, okay, like telmisartan, olmesartan, but those are basically they go and bind and block the angiotensin angiotensin 2 receptors. Okay. So, there are two types of drugs based on this angiotensin renin angiotensin system. One is the SE ACE inhibitors, and another is the angiotensin 2 receptor antagonist. Both are in the market now, and that is the major blockbuster drugs available in the market for management of hypertension. Okay, thank you.